Good afternoon and evening, everybody. I'm Randy Nesse, Director of the ASU Center for Evolution Medicine. My job is to offer the briefest of introductions, just to make sure you know that we are trying to create more of these evening and afternoon events so that we can welcome more members of our community. Anyone who's not on our mailing list, please go to our website and get there. We have lots of interesting things, and we welcome everyone. Our mission for the Center for Evolution Medicine is to establish evolutionary biology as a basic science for medicine and public health worldwide, and to thereby improve human health. And I can't think of any better example of basic science that has huge clinical implications and using evolutionary biology than today's speaker. But I'm not going to get to say anything about him because Melissa Wilson Sayers is the person who invited him. It's a wonderful example of cooperation. She's a member of our faculty on the center, and I'll turn it over to you, Melissa.
Thank you, Randy, and thank you everyone for coming. I am beyond thrilled that Ed Yong came to talk to all of us, and if you haven't bought his book, there's a table outside. Please do that. A reminder, if you can, scoot in a little bit. If there's an open spot in the middle, please scoot in because there will be more people coming in the next few minutes. Okay, and then the important part, which is that Ed said to keep his introduction short, and I've already talked too long, but I have 3 words prepared. So he has a wildly popular blog. He writes for The Atlantic. You should read everything that he's written because it will make you happy and more informed. But the 3 words are really that he's extremely intelligent, and he's able to distill things out of such a broad range of scientific disciplines that just amazes me that he's able to not only make this science accessible, but he puts a human aspect to it. And it invests you in the scientific process that's going on. I knew I would get so nervous for the second word, which is that he's extremely meticulous. So I know that when I see anything that Ed has written, it's right. I know that I can trust it. I know that he's not overselling anything. And I know that he's not underselling things. And so I think meticulous and diligent. And the last is that he's extremely genuine. So he cares about the scientific process, he cares about the people that are doing the science, and he cares about getting the science right. And sometimes that's something new and exciting, sometimes that's small incremental progress, sometimes it's not being reproducible. But there's always this genuine interest in making sure that what's presented is accurate and trustworthy. And so I am thrilled for this presentation and so excited that Ed is here. So please welcome Ed Yong.
When Melissa said that she only had 3 words to introduce me with, I thought that they were going to be like, and here's Ed. Thank you so much. That was really kind. It's an honor to be here. And what I'm going to do over the next sort of 40 minutes or so is give you a whistle-stop tour of many of the stories and themes that are in my book, I Contain Multitudes, an introduction to the microbiome, the microbes within us, and this grander view of life. And I'm going to start by taking you back in time by about 350 years. So the year is now 1675, and we are in Delft, which is a town in the Netherlands. This is the time of Vermeer, and one of his contemporaries, Anthony van Leeuwenhoek, is about to change the world. Leeuwenhoek is a draper and a civic servant. He is not a scientist. He's not a scholar of any kind. But he does have 2 very important qualities. 1, he is exceptionally curious about the world around him. And 2, he is the creator of the world's most powerful microscopes, or at least of the time. He makes them himself. He fashions very perfect spherical lenses, tiny but impossibly smooth, and he sandwiches them between 2 brass plates to create this object that looks a bit like a glorified door hinge, but is capable of magnifying things in the world around him 10 times better than anything else around. And Leeuwenhoek turns them onto everything else around, everything he can find, bits of animals, plants, and water. Water from the canals and lakes around Delft, water that collects in an urn near his house. And everywhere he looks, he sees tiny living organisms in extreme numbers. These are bacteria, microbes. And Leeuwenhoek has just become the first person in the entire history of the world, in the entire history of life, to see these things with his naked eye, to understand, to know that they exist. And that must have been a tremendous moment in the history of life on Earth. Leeuwenhoek also realizes that these living things abound in his own body because he takes dental plaques scraped from his own teeth and he views them under those same microscopes. And again, millions of little organisms swimming about, animated, clearly alive. Now, was he disgusted or revolted at the prospect of finding so much life in his body, realizing that there are more living things in his mouth than there are people in the entirety of the Netherlands? Actually, no. He was very excited. He was thrilled. Like, loads of living things in his mouth. How cool. He probably did not say. The idea that bacteria are things that we should fear, that we should be disgusted by, was that idea came later. It came when, centuries after Leeuwenhoek's discovery, other scientists realized that many of the most infamous plagues of humanity were the work of bacteria and other microbes. In quick succession, they were identified as the cause of plague, syphilis, gonorrhea, cholera, tuberculosis, leprosy, and many, many more. And so bacteria became forever associated with death and disease and dirt. They were things we had to destroy lest they destroy us first. And that framing of antagonism and villainy in the microbial world persists to this day. If you find bacteria on everyday objects, it is a sign that you have not cleaned properly, that you have done a poor job at destroying the microbial world. I deeply apologize to anyone with beards in the audience. I don't judge your life choices, and I don't see why Metro is doing that either. This view of life on Earth, I think, is deeply unfair. It's very misleading, because only a few hundred kinds of microbes are actually responsible for disease. The vast majority are, in fact, benign or beneficial, if not to us, then at least to life on Earth in general. And microbes are, in fact, the rulers of the planet. They have been around for the longest time. So if we take the full length of this screen to represent the history of life on Earth, so that the planet was created 4.5 billion years ago at this edge, and we are now here in the present moment, then humans evolved here. The dinosaurs probably arose between like here and, they ruled the world between here and here. And then multicellular life, so everything that you can see with your naked eye, arose around here. All the animals and plants you're familiar with there. So for the vast majority of life on Earth, from that point all the way back to its very beginnings, roughly around, I'm guessing here, it was entirely microbial. You would not have been able to see it with your naked eyes, which would have been fine because eyes only involved here. So, we are but the icing on a vast microbial cake. We, and by we I mean humans and the rest of the animal kingdom, the subjects of my book, evolved in a soup, in a sea of microbes. We evolved to live with them, to exploit the signals they provided into the world, and to crucially depend upon them. To this date, every animal, whether hawk or hummingbird, human, ant, elephant, whale, all of these things are teeming collectives of lots of different species. And I think the poet Walt Whitman said this best when he said, "I am large, I contain multitudes." We all do. Every human contains an estimated 39 trillion microbial cells in our body, roughly one for each of our actual human cells, which means that we are all just half the people who we think we are. The same is true if we look wider at the rest of the animal kingdom. This is a sponge among the simplest animals, just a couple of cell layers sandwiching a jelly-like filling. And it, too, contains a vast microbiome, sometimes so thick that under a microscope you can barely see the animal cells for the microbes that cover them. Ants live in colonies that can number in their millions, but every ant is a colony of millions in itself. A polar bear walking through the Arctic ice sheets looks completely alone, but is in fact surrounded. Bar-headed geese fly microbes over the tops of the Himalayas. Sperm whales dive them down into the deepest oceans. And when these gentlemen set foot on the moon, they were also taking a giant step for microbe kind. These bacteria, these other microbes, are not just stowaways. They're not just hitchhikers on our lives. They are also incredibly important. And in many cases, they bestow their host with almost unbelievable superpowers. This adorable creature is the Hawaiian bobtail squid. Very big on the screen. In real life, it would fit in the palm of my hand. It has luminescent, glowing bacteria in its undersides. And the light they project down from the squid matches the moonlight shining down on it from above, which means that predators looking from below cannot make out the animal's silhouette. Its bacteria cloak it in an invisibility cloak. These small white worms kill insect hosts by vomiting toxic bacteria into their bodies. This bird is the hoopoe. It paints its eggs with a bacterial paste that produces antibiotics that keep the chicks from contracting infections. This is the hyena. It has scent glands in its backside full of bacteria whose odors correlate with the animal's species, age, sex, and social status. It is effectively writing its autobiography in bacterial sense. And this is a flatworm called Paracatenula. It can regenerate almost its entire body, but as long as it has some of those dark-colored symbionts, those dark-colored bacteria living inside it. Chop almost everything off beyond the head, and as long as some of those bacteria remains, the rest of the worm will regrow, for reasons that we still don't understand. We humans, too, depend on the microbes that we carry for various aspects of our lives. We need them to digest our food, to provide us with nutrients, to train and calibrate our immune systems, to protect us from disease. I've been writing about this area of science for over 10 years now, and there are a few areas that excite me more, because I have always loved the natural world. I was always a fan of nature. I went to zoos a lot as a kid. Here is a picture of me with a kid on my back. And I watched David Attenborough documentaries. I read books about wildlife. And now I understand that whenever I go to a zoo, every resident and visitor in that zoo is a zoo in their own right. And that everything I can see with my own eyes is profoundly influenced by things I cannot see. And I know that I don't really understand my biology if I do not understand theirs and their contributions to my existence. Because when we look at our world through a microbial lens, even the most everyday and familiar parts of our lives become newly unfamiliar and wondrous. Here are several examples. I'm going to start with breastfeeding. Breastfeeding is a way of nourishing an infant, sure, but some 10% of breast milk consists of these complex sugars called human milk oligosaccharides, or HMOs, that babies cannot digest. They are there to nourish microbes in the baby's gut. And particular strains, like this one, B. infantis, which have evolved the right genetic tools to digest those sugars with maximal efficiency. And when it does so, it releases nutrients that nourish the baby's gut, it tightens up the lining of the gut, and it quenches inflammation. So a breastfeeding mother isn't just feeding her infant, but infantis as well. She's building an entire world. She's a sculptor of ecosystems, setting up these multitudes within her infant's gut. And those multitudes, in turn, set us up. All of us develop from a single fertilized egg into a fully adult body comprising trillions of cells. But that process of development doesn't unfold simply through the instructions encoded within our own genomes. Oftentimes, it also requires bacterial assistance. These animals are tube worms. Small, they construct these little white tubes that, although tiny, can become so dense that they clog the propellers of ships. They're a particular problem in Pearl Harbor, for instance. These tubes only form when the worms become adults, and the worms only become adults when they encounter the right bacteria in their environment. Those microbes are necessary for their metamorphosis, for their completion of their life cycle. If for some reason all the microbes in the sea were to vanish, these worms would probably go extinct within a generation. And the same is true for many other marine invertebrates. They too metamorphose upon contacting bacterial signals. Many other animals that we are more familiar with also rely on constant bacterial signaling throughout our lives in order to shape our development. Here are two of them. The one on the right is loaded with microbes, and the one on the left is not. This is a germ-free mouse. It has been completely disconnected from the microbial world throughout its life. It was raised in one of the sterile plastic isolators, and it ate sterile food, drank sterile water. It does not contain multitudes. It is one of the the strangest animals on the planet because it has never encountered a bacterium in its life. And as a result, it has many physiological woes. Its bones don't develop properly, its blood vessels, its gut. Loads of organ systems in its body are incomplete because it has not encountered bacterial signals as it has been developing. So, we see that microbes actually help to shape and sculpt the bodies of their hosts. Far from just being destroyers of life, they are actually its constructors. They also help to safeguard the health of their hosts once their bodies have been built. The immune system is something that many of us think of as this militaristic force, as a defense force that patrols the body and eliminates anything that is foreign, other, or non-self. That metaphor is at worst wrong and at best misleading, because we see that different classes of microbes can actually stimulate the development of classes of immune cells. They help to build the immune system, and they also calibrate it. They help to set the right balance between arms that trigger inflammation, that allow us to fight off infections, with arms that reduce inflammation, that stop us from overreacting to harmless and imagined threats in the world around us, from dust to pollen to allergens in our food. And in turn, that community of microbes, that immune system which has been built and tuned by microbes, helps to select for the microbiome, that they decide which microbes get to live in our bodies and where they get to persist. So I think of the immune system less as that and more like this, a team of rangers in a national park, whose job it is to care for the species that live in the park, to keep their population's levels under control, and to chuck out any invaders. We also see that the microbiome can affect the way we behave, can affect our brain. We know this from a lot of rodent studies in which, depending on the presence of microbes or the specific strains or species that live within the animals, you see differences in their susceptibility to anxiety, their resilience to stress, aspects of their mood, their personality. The extent to which that applies to us humans is still being investigated, but I think it is plausible, perhaps even probable, that the microbes within us can influence our brains and behavior too. Even though we think of ourselves quite narcissistically as rulers of our own fate and our own thoughts, we also have a vagus nerve that connects our gut and our brains. The immune system sends signals between those two organs, and the bacteria in our bodies also create serotonin and dopamine and other chemicals that we think of as brain signaling chemicals. So thought, digestion, development, immunity, health, all of these aspects of our lives that we think of as the province of individuals are really the result of partnerships, of conversations and negotiations between us and the microscopic symbionts that we carry. And those partnerships matter over an individual's lifespan, but they also matter very deeply over longer evolutionary timescales. So as we see time and again, microbes influence the evolution of their hosts. They open up opportunities and lifestyles that would have been completely denied to their hosts. And in doing so, they pave the way for tremendous evolutionary success. Here is an example. These are hemipterans, sap-sucking bugs, an aphid, a cicada, and a leafhopper. This is a group of insects that has made a living from drinking the sap of plants. And that sap is lacking in certain essential nutrients, like some amino acids. These insects can cope with that shortfall because they have bacteria living inside their cells which act as dietary supplements, which help to manufacture the nutrients that are missing from their meals. And without those microbes, this group would never have been able to tap into this incredible food source that is so widespread and common. They would never have been able to succeed against their fundamental animal nature because they cannot manufacture said amino acids on their own. And there are now some 85,000 species of these animals, a great example of animal success founded on bacterial ability. In the deep oceans, there is an even better example of this supplementation taken to a greater, more extreme degree. These are tube worms, like the ones I showed you before, but meters in length. And they have these beautiful red-plumed gills, which look like tubes of lipstick that have been pushed out all the way. They live in hydrothermal vents, habitats where food is scarce, if it is present at all. But they have microbes in their bodies which use minerals in the vent environment to provide them with energy, chemosynthetic life, rather than the photosynthesis that we are familiar with on the surface of the planet. And their bacteria provide them with so much energy, in fact, all the energy that they need, that these worms and many other creatures of the vent environment have lost their mouths, their guts, and their anus. They have no digestive tract, because why bother eating when you have microbes to provide you with all the energy that you need? Many other animals, of course, have kept their guts and their digestive tracts and have expanded them into vast fermenters, like apartment complexes, to house the microbes within them and allow them to get on with the digestive work. Wildebeest and many other grazing animals cannot digest the vast majority of the carbohydrates in the plants that they eat. They rely on microbes in order to perform those acts of biochemical wizardry. And in return, their bacteria provide them with something like 70% of their energy budget. Again, wipe out all the microbes in the world, and these plains of Africa would be silent and barren, or at least to our eyes. There would be plenty of bacteria there, they wouldn't care. Okay, so we've seen that microbes influence the evolutionary opportunities that different groups of animals can take, allowing them to succeed against their basic nature. They can also accelerate the pace of animal evolution by allowing us to adapt to new challenges very, very quickly. So rather than, say, trying to fill a skills gap in our organization by training our existing staff to have those skills, we can just recruit new members that have those skills themselves. This is the Japanese beanbug. It does something very similar. It can become instantly resistant to insecticides that are sprayed upon it by swallowing microbes in the soil that can detoxify those chemicals. It can instantly acquire the adaptations that the bacteria themselves had to gradually acquire just with a meal. The same applies to this adorable desert wood rat. It can also become instantly immune to toxins of one of the most common plants in its desert home, again by eating the right microbes, either that live on those plants or that are found in the stool of other wood rats that have those same abilities. These insects, aphids, can trade bacteria that protect them from parasitic wasps, and they do so through sex, making this one of the rare cases in the world of a desirable venereal infection. Yeah. Okay, so, one more story. Humans can also benefit from these kinds of transfers too. So this is one of my favorite microbiome stories. It involves seaweed. Seaweed has been harvested off the coast of Japan for many centuries and used to make nori, which is an ingredient that we still use in sushi. Nori today is cooked, but in the past it used to be eaten raw. And in doing so, people swallowed marine microbes that were found on the seaweed and were exceptionally good at digesting the rare carbohydrates found on these meals. And when those microbes entered our guts, they did something that microbes do all the time, but that is extraordinary to us. They swapped genes. Humans can only pass our DNA down from parent to child, but bacteria and other microbes can often do so horizontally, trading DNA and genetic material as easily as you or I might swap phone numbers or ideas. And that's what happened in the guts of those olden-day Japanese eaters. The marine microbes and the seaweed they swallowed passed on genes for digesting the carbohydrates in those seaweeds to the microbes in their gut, which is why to this date, the gut microbiomes of Japanese people are uniquely qualified to digest the carbohydrates found in nori and other sushi-like ingredients. Okay, so microbes are cool, microbes are important. But one thing I want to stress is that microbes are not our friends or our enemies. They are not good or bad for us. They are simply there. We are just another habitat in which they live, just like a piece of soil or a drop of water. It is their world after all, and we are just living in it. And I stress this because that old narrative about bacteria as germs, as enemies that we needed to kill, has been replaced a little bit by a narrative of friendly bacteria and good microbes. And I feel that that is just as wrong. In reality, the microbes that share our lives are very contextual. They can be both allies and enemies. Those in our gut can help to digest our food and train our immune system. But if they cross the lining of the gut, they can enter our bloodstream and cause inflammation and sepsis. The very same microbes, for the matter of a millimeter or so, can turn from allies to enemies. And throughout the natural world, we see that every instance of partnership is tinged with conflict, where the interests of both partners are never fully aligned. And we see this in the microbiome as well, which is why I always say that there is no such thing as a good microbe or a bad microbe. Their relationships with us are very dynamic, and we need ways of containing our multitudes, of keeping them in check. We use physical barriers, like a great wall of mucus in our gut that keeps them in place. We have our immune system to keep them in line too. We feed them with specific foods in order to select for the right species, much like the HMOs in breast milk nourish B. infantis above all others. And when those means of control falter, as they often do, then our relationships with our microbial partners can break down, can go awry. You see some of these dynamics at work in coral reefs. Corals are animals too, and they too have teeming microbiomes. And many of the things, corals also have rivals. They have turf algae, kind of scummy green seaweedy things that compete with them for space and nutrients. And many of the things that kill corals do so by tipping the balance of this competition, giving the algae the seaweed an advantage. And when they overgrow, they release dissolved organic carbon into the water, essentially dissolved sugars, fast food that nourishes specific microbes that live in the coral's microbiome. And it seems that those that are more likely to cause disease are more likely to bloom as a result. And so you get this entire shift in the corals community away from a healthy one towards one that is more likely to make them sick. It is a very different view of microbial disease than what we're used to when you typically have one microbe, say like Mycobacterium tuberculosis, causing one disease, tuberculosis. Here is a case where you have an entire community that has shifted into a disease state, a pathogenic state. And that is something that is commonly called dysbiosis. You see examples of this in the human body too. There is a growing, almost absurd list of conditions that have been linked, correlated to changes in the microbiome. And in many cases, it is still unclear if it's the change in the microbiome that leads to disease, if it is a consequence of disease, if it is both or neither. Well, there are studies that hint at a more causal effect rather than just providing correlative evidence. And many of those involve those mice that I showed you earlier. So you can do transplant experiments where you take a rodent's microbiome and implant it into one of these animals. And often when you do that and the donor has some kind of illness, whether it's diabetes or heart disease or colorectal cancer or obesity or whatever, you see that many of the symptoms transfer across to the recipient. So that suggests that to an extent, the microbes are not just going along for a ride, they are also grabbing the wheel. Now, what that means for human health, I think, is still being worked out. It's subject of a lot of investigation and a lot of good work. But we still don't know the extent to which these mass experiments apply to us in the wider world, the extent to which the changes in the microbe, how strongly they are affecting our health. And we're also looking at what's changing the microbiome in the first place. So for us humans, there are many possible answers to that. We live in a different world than we used to, a world of urban cities rather than muddy soils and countrysides in close contact with a large variety of animals and microbes that live within those environments. We sanitize the world around us, assaulting it with antibacterial everythings. When I was reporting for the book, I picked up a handset in a hotel I was staying in, and it said antibacterial handset. I don't know how you make a handset antibacterial, but apparently there was one. Antibiotics are drugs that we use to kill microbes. We have done so with tremendous success, and we have saved countless lives in this way. We have controlled many important infectious diseases. But these drugs also come with collateral damage. They harm the microbes that we rely upon, as well as those that might cause us harm. And over time, our diets have changed. We eat less plant food, less fiber than we used to. Fiber nourishes a wide range of microbes in our bodies. It contains a wide range of complex carbohydrates, and no single species has the right cutlery set for digesting all of these. So fiber nourishes a wide diversity of microbes, and if we reduce the amount of plants in our food, that diversity starts collapsing. So, is there anything we can do about this? In theory, yes. As I have shown you, microbiomes can change in very positive and very negative ways very quickly. And that gets people's attention because it means that maybe we can change them in deliberate ways that we choose, ways that we design with equal ease and speed. But in practice, these sorts of changes are very hard to execute because microbial medicine is very complicated. Remember that we're dealing with complex ecosystems here. So consider something like probiotics. Probiotics have a long history. We have sachets and capsules and drinks and yogurts that supposedly contain beneficial microbes and that have been linked to a vast array of health claims. And yet, even though these products seem to be very good for cases of infectious diarrhea and a few other minor conditions, they don't live up to the hype that surrounds them. They end up being very medically underwhelming. And there's probably good reason for that. These are products that contain small numbers of bacteria, orders of magnitude lower than the vast multitudes that live within us. Those strains were often chosen for historical reasons because they're easier to package and manufacture rather than because they are the A-listers of the gut and are very good at colonizing it. And they also are often industrialized strains, domesticated bacteria that have been grown in proprietary cultures for a very long time. So swallowing one of these things is a bit like taking captive reared animals that have never been released into the wild and shoving them into a jungle in very small numbers. And it should come as no surprise that they just don't really fare very well. They don't manage to colonize the body with any degree of success. I also compare them to a breeze that blows between two open windows and that might rattle some objects along the way, but that does not have a lasting impact. So maybe instead of this approach, we're better off at giving people vast communities of bacteria and other microbes, and particularly those that are well suited to life within us, that are well adapted to our particular ecosystems. And that is certainly the idea, the logic behind a somewhat unorthodox treatment called the fecal transplant. Fecal transplants are exactly what they sound like, and let me tell you that it is not easy to find a good image to illustrate. So I've prepared a very technical explanation for how fecal transplants work. Pay attention. You have a sick person, and you give them stool from a healthy person, and they get better. Okay. So, I also think of fecal transplants as a bit like an ecosystem transplant, much like returfing a lawn that has been overrun with dandelions with fresh new pasture. Now we'll leave you on this slide because it's pretty. The weed, in this case, the dandelion in this metaphor, is typically a bacterium called Clostridium difficile, or C. diff, which is incredibly invasive. It causes severe, intractable, and sometimes fatal cases of diarrhea. And fecal transplants have proven to be extraordinarily good at treating C. diff. In one famous Dutch clinical trial, patients with C. diff who received vancomycin, a standard antibiotic, were cured about 27% of the time, whereas those who received fecal transplants were cured about 94% of the time. The trial was stopped early because it became clear that it was unethical to give people the antibiotic rather than the crap. So, on the back of that success, people have tried fecal transplants for all sorts of other conditions, many of the ones that I've been telling you about. But there, the success rates are lower and certainly more inconsistent. So even with this treatment, it is difficult to reset a microbial ecosystem. Perhaps there's something unique about gut with C. diff. Perhaps it's a low-hanging fruit that is easy to reinstall a donor stool into. But really what this tells us is that even with something as seemingly promising as a fecal transplant, we still don't really fully understand why they work. What is it about them that is curative, even in this case? And then how would you duplicate it? Many people have tried. There are companies and scientists who've tried to create cocktails of microbes that are found in stool and that can be administered to people without having the ick factor. And these fake poop pills have been tried in a few trials. And the most promising candidate that seemed to work in initial phase 1 trials recently flunked out of a phase 2. Disappointing, yeah, but it did allow me to write this headline, which I might retire on. You thought you were hearing about the microbiome instead. It's just half an hour of dad jokes. Okay, so the idea behind probiotics in fecal transplants is very sound. But these are complicated things that we're trying to do here. This isn't just a case of, say, having a vitamin deficiency, which you can plug by giving people supplements or making them eat an orange or whatever. This is a complicated problem. It is a problem of ecosystem engineering, of trying to control something as complicated as a coral reef or a grassland or a jungle, with all the complexities that entails. And I think we are still at a very early stage of trying to understand these worlds inside us. We still don't have a full membership list. There are still many unidentified parts of our microbiome. We still don't really understand the variations between us. Our microbiomes are very different. Mine is different to yours, and yours, and yours. And we can only explain maybe about 10% of that population-wide variation, despite everything that we've learned about diet and antibiotics and all of the above. And if we go out into the wider world, our ignorance gets even deeper. So here is a tree of life that was published quite recently. This is a pictorial depiction of everything that exists. And I want to just highlight the bottom half of this, and that little green plume down there, that little twig on that little plume is not only you, but all the animals and plants and all the life that you know of is just here. And the rest of this thing is microbial. And if you look at all the red dots there, those are groups that have never been seen or isolated. We only know about them by sequencing their DNA. And that purple spray on the right was only discovered, I think, within the last year. So I've been giving this talk a year ago. None of that vast swathe of bacterial diversity we would know about. I'm going to deviate from my usual talk a little bit by pointing out that one of those groups has actually, members of one particular phylum there has actually been seen. I saw a poster at a conference just a couple of days ago. It seems to be an epibiont, a bacterium that lives on other bacteria, harmlessly at first, and then parasitically over the course of its growth. So, oh my God, we know nothing about the tree of life. Like, really, really nothing. And I think that makes this area of science deeply exciting. That there is still so much to learn, so much to play for. So I'm going to finish this talk by giving you one last story about how the exploration of the microbial world, with no particular end game in mind, led to some really cool and unexpected and significant benefits for us. And it involves this guy. He's called Simeon Bert Warbach, and in 1924, he discovered a new bacterium living inside the cells of insects. And he and his partner, Marshall Hertig, had no idea what this thing was, or whether it was common or not, what it did. It actually took them 12 years to even give the damn thing a name. And Hertig named it after his friend, and he called it Wolbachia. Wolbachia, several decades later, turned out to be everywhere. Scientists who were studying insects and the bacteria they contained suddenly realized that they had all been independently studying the same thing, which was this thing. This thing is in 40% of species of insects and other arthropods, and given that they themselves are the most abundant and diverse and species-rich groups of animals on the planet, that probably makes Wolbachia one of the most successful bacterium in the world, one of the greatest pandemics in the history of life. It infects a variety of hosts, and what it does to those hosts varies from species to species. Wolbachia doesn't really like males because it passes down the female line from mother to daughter. So if it gets into sons, that is a dead end to it, and it deals with that in many ways. In this animal, the spectacular blue moon butterfly, it sometimes just kills male embryos outright, giving adult populations that can have 100 females for every male. In some wood lice, it feminizes the males, transforming them into females as they develop. In some parasitic wasps, it allows the females to reproduce asexually by cloning themselves, so they have no need for males whatsoever. Many people are smiling in the audience. Wolbachia can also be a mutualist, an ally. This is the bedbug, a blood-sucking insect, and Wolbachia provides it with B vitamins that are missing from its blood meals. This red leaf has a little green island at the bottom of it. That is the work of a caterpillar called the spotted tentiform leaf miner. It feeds on leaves, and using Wolbachia, it stops the progress of the fall, stopping the leaves from yellowing so that it can continue eating within those green islands for as long as possible. But the insect host that most concerns us for this story is this one. This is Aedes aegypti, the tiger mosquito, the insect that spreads yellow fever, chikungunya, dengue fever, and a little thing that you may have heard of called Zika. For about 25 to 30 years, Australian scientists have been trying to shove Wolbachia into this insect, one of the few that it doesn't naturally infect. Why? Because in this insect, Wolbachia stops it from transmitting the viruses behind those diseases. It turns it from a vector into a dead end. And because Wolbachia is so good at manipulating the reproductive biology of its hosts, within a few generations, it is very good at spreading through wild populations. So the idea is that if you release a reasonably small number of these infected mosquitoes, within a few generations of their time, months to us, the entire local wild population should contain Wolbachia and thus be unable to vector these important diseases. And that approach has been tried in a few suburbs in Australia. This was done in 2011. From January to May, at the end of this graph, these grey bars represent the release of Wolbachia-infected mosquitoes with a brief pause when they got hit by a cyclone. They're Australians, they don't care. And this green line represents the proportion of Wolbachia in the wild population. And you can see that very quickly it goes from 0 to almost 100%. Following this success, the scientists who've been spearheading this approach have gone global. They've gone to tropical megacities in Brazil, Colombia, Indonesia, Vietnam, and Australia also. And they are testing this approach in places containing millions of people to see if it can really reduce the incidence and spread of these important illnesses. And this approach, I think, has many, many advantages to it. It is not toxic, unlike, say, insecticides. And unlike insecticides, which must be resprayed continuously, once you release the Wolbachia-infected mosquitoes, you're off to the races. They can sustain themselves. The approach is also involves no genetic modification. So for people who have a problem with that, it makes an easier public sell. The story of Wolbachia and mosquitoes, to me, is the story of animal-associated microbes in a nutshell. These things were neglected at first. Wolbach and Hurtig could not possibly have predicted that the thing that they discovered had the potential to save hundreds of millions of lives. In fact, Wolbach died in the 50s before anyone realized how common Wolbachia was, before he realized that his very name had been grafted onto one of the greatest pandemics in the history of the world. And I think we have neglected microbes for the longest time, and then we feared them. But now I think it is time to appreciate them. Only recently have we understood the important roles they play in our lives and that of every other animal. Only recently have we begun to be able to manipulate these partnerships in an attempt to improve our health and those of other species. And though our attempts at doing so are clumsy and a bit fumbling right now, the potential for this area of science is enormous. And certainly, it changes our understanding of the view around us, the way we see ourselves and our place in the world. And I think it all begins with basic curiosity about the world. That's what led Wolbach and Hurtig to discover Wolbachia. It's what led Leeuwenhoek to turn his microscopes on a drop of water. What a weird thing to do. Why would someone do that? And yet he did, and opened the doors to this incredible new world. And it's that type of curiosity and desire to learn more about the things that we share our lives with. That's why I wrote I Can Tame Multitudes. Thank you very much.
So we'll just ask that you use the microphone because it's being recorded and other people can access this later.
Thank you. I understand that most microbes haven't been identified in the wild yet or called out. And my question for you is, first of all, do they have to be called out individually before we can use them, or can they be used as part of a community? And secondly, if they're going to be found out, is that more likely because they're easy to find or because they take part in a significant biological interaction?
That is a good question. Do we even need to find them? I mean, I would argue yes. I think it's basic biogeography, like trying to work out who is where. And I think we won't get to like the deeper questions, like how they interact with each other, how they interact with us. We won't be able to use them unless we discover who they are. And I don't mean like culturing them, looking at them under a microscope. I just mean like even working out their genes. Like that is how most of these things have been identified and named. And I think that is that's the start. In the start of the book, in the introduction, I compare this era of microbiology to a time when like adventure naturalists like Wallace and Darwin was sailing around the world, grabbing new species, like cataloging the sheer diversity of flora and fauna on all these islands. And we are the islands now, and the people who are doing that exploration are microbiologists. They're the ones who have, swapped nets and forceps and ships for trips to the zoo and cotton swabs and sequences. And that's what it looks like, I think, now to explore the microbial world. And I think that it's still a worthy thing to do.
So I've heard there's now companies that offer to screen or test your microbiome or whatever. Mm-hmm. What kind of information are they getting, and is it of any real value?
Yeah, that's a really good question. And I think the answer depends on, so value to whom? So obviously, as a consumer, you are paying for that service, and you are getting a view back about your microbiome. My contention is that if you are doing that to further research, if you are doing it as as a sort of citizen science isn't the right word, but if you want to take part in it and increase the N of whatever future microbiome study is going to come out by 1, then that's great. If you're doing it for like actionable advice on your own body and health, I think that we're still at too early a stage to do that. I don't feel that these services provide people with something that really tells them about the world inside them in ways that can actually inform their life choices yet. I think it's still way too early. Like even 23andMe and services like that, which are based on our own genomes and are much more mature have a longer history of development and are contributing very heavily to research, even that tells, I've done that and I gain a kind of limited understanding, extra understanding of myself in that way. I know that it told me that I'm 99% likely to be of East Asian origin, which came as a total surprise. I had no idea. I mean, blew my mind. I know that I have dry earwax, and now you all do too. I feel that I wouldn't recommend it as a service, and I certainly haven't done it. People have asked me, when you wrote the book, did you get your microbiome sequenced? And the answer is no. I also think that it varies so much. It's such a dynamic entity. It changes over a 24-hour cycle with every meal, with drugs I take. So, yeah.
Do you take a probiotic?
I do not. I eat lots of yogurt, but because I like the taste of yogurt. I feel that is an underappreciated reason for people to eat food nowadays.
Which leads me to my question of what has your research done to change any of your daily habits, and what suggestions would you bring it to a consumer level from your research?
Yeah, that's a good question, again. And I feel that I have... so my diet hasn't changed. I am a vegetarian, and I was before I started doing this. Actually, no, wait, that's not true. I'm a vegetarian before the book came out, but I started becoming a vegetarian afterwards. And it wasn't because of this, even though what the science of the microbiome confirms that eating a wide variety of plant foods and fiber is important. But I feel that there are many reasons why people make the dietary choices they do, and maybe this is part of it, but certainly eat more fiber isn't new advice. It's not it's something that we've been told for a very long time, and it's confirmatory, but not revelatory. I have not made more specific dietary changes than that, because I think that the science is still in its infancy, and so that we're not in a place to extrapolate from small-scale studies what people as a general population should do. And as a rule, I generally find that with dietary advice from studies like this, the more specific you get, the more likely it is to be wrong, which is why I think eat fiber seems to be a good, broad thing that people can do. I certainly feel less, I was never one to really use a lot of antibacterials or things like that, but I certainly feel more comfortable with that stance. I think having less fear about the microbial world, and even I've been writing about this for a while, cannot help but associate these things with dirt, and trying to actively break that mindset has been one thing. But I am still dubious that this is a thing that can give, this is an area of science that at the moment is capable of giving people actionable personal advice. I feel that the thing that the writing I Contain Multitudes has done for me, above all else, is change my perspective on the world around me and the way I see nature, my connection to the world. Every time I go to a zoo and I shake hands with people, all of that takes on this new light, and I like that.
So it's now 1 minute until 6 o'clock, and there were many hands here. And what I'd rather do, rather than choose one last person, is to take the time to thank Ed. Everyone, please.